Before we talk about red cell indices specifically, we're going to talk a little bit about the different appearance of red cells in our various veterinary species. And just to realize that red cell indices, when we talk about them, we're really going to be talking about size or volume of red cells, as well as hemoglobin content. And so we also assess not just these indices, but how the red cells actually look. So that's why I'm going to kind of go through this. The top left picture is a dog, and the dog has central pallor because the red cells are a biconcave disc, and they're kind of our standard size that we compare everyone else to. Cats, which is the middle top, they have smaller red cells which lack central pallor, which are very similar to horses, which are also smaller red cells which lack central pallor, and horses are on the top right. Cows are similar to dogs in that they have central pallor, but the cells are a little bit smaller. And for that matter, goats and sheep, different types of ruminants, also have very minimal central pallor, but their red cells are very, very small. They're some of the smallest red blood cells. Camelids have elliptical red blood cells, which is very interesting. It just means that it's hard to do some of the standard blood assessments that we do on hematology analyzers because of the shape, and that's true of birds and reptiles as well, who have the weird shape and that are nucleated. The two indices we're going to talk about are MCV and MCHC. And MCV stands for the mean cell volume. And this is essentially just what it describes. It's a measure of the volume of a cell, of the average of all of the red blood cells, because it's the mean. The MCHC is the mean cell hemoglobin content. I'm going to abbreviate hemoglobin. And so that's just a measure of the hemoglobin content of the average of all of the red blood cells. Again, it's a mean. MCHC is similar to MCH, but it doesn't have, it's not as useful, so we don't really use MCH. So we're actually going to mostly ignore that when we look at our CBCs. So we don't tend to evaluate our indices first. We actually look at regenerative status first, again, in everybody but the horse. Although when we characterize an anemia, we characterize it earlier. It's just that we don't really use the MCV and MCHC to figure out the etiology except for occasionally. So first we might look at total protein, we might look at regenerative status, and in that we'll characterize our MCV and our MCHC. So now I'm going to actually go through a little bit about characterizing these and telling you a little bit where they come from. So with instrument, so if you do a CBC on a hematology instrument, some measures are going to be measured and some of the analytes are actually going to be calculated. So what is measured, meaning directly measured by the instrument, what we talked about before, our red cell count in millions, our hemoglobin concentration, as well as our MCV. That is also measured. What's calculated are numbers that we actually calculate from these, and so we calculate our hematocrit, and that's actually you get from multiplying your red cell count in millions from your hematocrit. It's a little bit different, but you can get it that way. And your MCHC, which again is about hemoglobin, so it's calculated from your hemoglobin that's measured. And this is relevant for things that we'll talk about later. So we'll start with MCV. So again, MCV is the mean cell volume, so the size, the volume of the cell. So when you see an increased MCV, meaning it's greater than the reference range, we are going to call that macrocytic. If it's within reference range, we're going to call it normocytic. And if it's decreased below the reference range, we're going to call it microcytic. So a general cause of a macrocytic anemia is one that is regenerative, where you have accelerated erythropoiesis resulting in a skip division in larger cells, whereas a general differential for a microcytic anemia could be iron deficiency, where you have less hemoglobin, which results in an extra division in erythropoiesis in a smaller cell. So that's kind of on the blood smear, or excuse me, on the CBC part of it. We can also assess MCV sort of on a blood smear. It's not quite as accurate, but we can look for things called macrocytes and microcytes. And these are, by definition, larger cells and then smaller cells. So, of course, you have to know the normal cell size for the animal. And so you may see occasional macrocytes or occasional microcytes depending on the respective disease process, but because there aren't enough numbers of cells that are affected, it may not impact your MCV or your your MCV to make it increased or decreased. All right, again, so MCHC has everything to do with the amount of hemoglobin in all of the red cells, what the average is. I'm going to skip increased MCHC for a second, but normal MCHC is normochromic. Decreased MCHC is hypochromic. And the problem with increased MCHC is you can't have extra hemoglobin within the cell. So this is hyperchromic, but we don't really classify it. So we don't have a name for that. Things that cause it are free hemoglobin, either intravascular hemolysis or in vitro hemolysis, Heinz bodies, there's spectral interference with hemoglobin, with lipemia. We'll talk about that when we talk about hemolysis. We'll talk about increases in MCHC and why they're invalid. Now, when you look at a blood smear, it's similar to judging size. You can look at how much hemoglobin they have, and so you can tell if they appear hypochromic. So you might say that there's hypochromasia. Again, we don't see extra hemoglobin. So a common cause of decreased hemoglobin would be an animal that has a regenerative anemia who has a lot of reticulocytes that are large but have less hemoglobin per volume. Iron deficiency is another differential where you have small pale red cells, but that's less common. So admittedly, a lot of the anemias we see are not very exciting, and they're what we call normocytic normochromic, kind of right in the middle. So not very exciting. Classic patterns we will see. So you're always going to actually look, you're going to see there's anemia, and then you're going to determine if it's regenerative or non-regenerative if you have the ability either from polychromasia or reticulocytes in the appropriate species. But again, a regenerative anemia tends to be larger, paler cells. So they'll be hypochromic but macrocytic, so that's more common. And it's very characteristic of regenerative anemia, where iron deficient cells tend to be very small and very pale. And so that's kind of the two classic features. Something else interesting that we can see, you can see Japanese breed dogs. They have a low MCV, and dogs with portosystemic shunts can have a low MCV, be microcytic, but they're normochromic. We're going to practice this a lot. So when you're asked to characterize an anemia, you'll give the degree of the anemia, which I'll tell you about in another lecture. That's something that takes some practice. Then you'll say whether it's regenerative or not, except in the horse, and then you'll describe the indices. Is it macrocytic, microcytic, normocytic, etc.? And so it'll be, for example, regenerative anemia might be regenerative, macrocytic, hypochromic.